Okay, so think about this. You find one amazing, rare plant that could be a breakthrough medicine. How in the world do you get enough of it to help millions of people? Well, it's not magic, that's for sure. It's an incredibly high-stakes scientific process. And today, we're going to dive deep into that journey, from one perfect plant to a massive global supply. So when you see a field like this, don't just think, oh, pretty flowers. No, no. What you're looking at is basically a massive outdoor laboratory. Every single plant here has been genetically selected, meticulously cultivated, all for the specific medicinal compounds it creates. This is precision agriculture on a whole other level. Which brings us to the billion-dollar question, right? You found it, your super plant. It's perfect. So how do you make a million more that are exactly like it? It's definitely not as simple as just planting a bunch of seeds. Turns out, there are three main scientific paths you can go down. All right, let's look at path number 1, the natural clones. The best way to think about this? It's like nature's very own photocopy machine. Seriously, plants have been doing this for millions of years, and we've just gotten smart enough to harness it. The technical term for this is vegetative propagation, and it's pretty wild. Instead of using a seed, you take a piece of the parent plant. Could be a stem, a root, even just a leaf, and you grow a whole new plant from it. The new plant isn't just similar, it's a perfect genetic clone. And you've probably seen this happen right in your own kitchen without even thinking about it. Ever planted a clove of garlic? It grows into a whole new plant, right? Or those little eyes on a potato that start to sprout? Yep, those are just clones waiting to happen. The plant is basically using its own pantry as a starter kit. And wow, have plants evolved some clever ways to pull this off. Ginger, for example, spreads using this underground stem called a rhizome. It just keeps sending up new shoots as it goes. And peppermint? It sends out these runners that literally race across the top of the soil, putting down roots and starting new plants all over the place. But here's the catch. There's a really critical trade-off.
On one hand, cloning is awesome. It's fast, it's consistent, perfect for medicine production. The downside? You get a monoculture. Every single plant is genetically identical. And that means if one new disease comes along, it could wipe out the entire crop. Think of the Irish potato famine. That's the risk you run.
And that risk, well, it leads us right into path number 2. So if cloning is the photocopier machine, this next method is more like a genetic lottery. It's all about shuffling the genetic deck and seeing what new combinations you get. This is what we call sexual propagation, which is really just a fancy way of saying good old-fashioned growing from a seed. Every single seed, like this bean here, is a totally unique mix of its parents' genes. It might get all the best traits, or, it could be a complete dud. That's the lottery part. But here's the thing about seeds. They're smart. They have these built-in survival instincts. They're not just going to sprout the second they get a little wet. Nope. A lot of them go into this deep sleep called dormancy, and they wait for the absolute perfect conditions. So to wake them up, we basically have to trick them. We might scratch their tough outer shell, soak them, or even stick them in the fridge to fake a winter. Now, the payoff for playing this genetic lottery can be massive. This is exactly how we breed new, stronger plants that can fight off diseases. But it is a long game. It takes way more time than cloning, and you just never know for sure what you're going to end up with. Okay, so we've got the reliable photocopy method, and we've got the high-risk, high-reward genetic lottery. So what's path 3? Well, this is where science fiction becomes reality. This is how you grow thousands upon thousands of plants, starting from just a single cell in a lab. The magic word here is totipotency, and honestly, you should think of it as a plant cell's ultimate superpower. It's incredible. Unlike most animal cells, a single mature plant cell has the amazing ability to turn back the clock and grow into a completely new plant. We're talking roots, leaves, the whole shebang. So how does this actually work? Well, step 1, scientists take a teeny tiny piece of the original plant. It's called an explant. And this has to be done in a super sterile, clean room environment because any contamination could ruin the whole thing. Next, that tiny explant gets placed into this special, nutrient-packed gel. It starts to multiply like crazy, forming this blob-like mass of cells called a callus. Then, and this is the really cool part, by adding specific plant hormones to the gel, scientists can tell that callus, hey, it's time to start growing some shoots. Then they switch up the recipe, adding a different cocktail of hormones that tells those new shoots to start growing roots. And would you look at that? You get dozens of perfect, tiny little plantlets all growing in a single jar. And you can scale this up to make millions of identical clones. And this stuff right here is the secret sauce. It's called a culture medium, and it's basically a 5-star nutrient soup for baby plants. It's got everything they could possibly need, all the right minerals, vitamins, even sugar for energy. But the real key ingredients are the growth regulators. Those are the hormones that act like a director, telling the cells exactly what to become. But you know what? Growing millions of perfect plants is only half the battle. Now comes the really tricky part. You have to harvest it at the exact right moment. The moment when its medicinal compounds are at their absolute peak. And in this game, timing is everything. See, a plant's potency isn't constant. It changes all year long. For instance, you want to harvest roots and rhizomes in the autumn, right after they've spent all summer storing up energy for the winter. But bark? You harvest that in the spring, when the sap is flowing and it peels off the tree easily. It all has to be perfectly timed. And the harvesting process itself is, wow, it's about as strict as a surgical procedure. The World Health Organization has a whole list of guidelines. You have to harvest when it's dry, never wet. The tools have to be spotless. The plants can't even touch the soil. It is all about preventing any kind of contamination and protecting that precious product. Okay, so the plant is harvested, but now a new clock starts ticking, and it's ticking fast. The moment a plant is picked, enzymes inside it start to break down all those valuable medicinal compounds. You have to move quickly to lock in that potency. So how do you stop that breakdown? Well, the most common way is just drying. The goal is to get the water content down to about 10%. Why? Because that basically puts those destructive enzymes to sleep. You can do this the old-fashioned way, just air drying, or use high-tech stuff like special ovens or even spray dryers for extracts. And this is what it all leads to. This is the final product. These carefully dried, sorted, and preserved plant parts are what the industry calls crude drugs. They're finally stable, they're potent, and they're ready to be transformed into the medicines that so many of us rely on. So, from a single cell in a lab all the way to a carefully preserved remedy on a shelf, every single step is this amazing blend of nature's potential and incredible human precision. It's a journey that really makes you appreciate what it takes to unlock the healing power hidden inside these plants.